computer. Yay. Red dot's blinking. My neurons might turn on any minute now. Oh. So instead of building a bear, today we're going to build a brain. And hopefully we get about halfway through this chapter today. And it's Thursday morning and it's a gray day outside. You know, I wish I had like a little blanket around me and I could just sort of sit here and mumble for a couple of hours. But I'm going to try and introduce the process by which the brain is actually built. And in utero, around 3 to 4 weeks, you actually have 3 major sections of the brain in place. Like, why do I need to use these words? You don't really. You're never going to see prosencephalon or mesencephalon or rhombencephalon, not in an exam in this class. But what you do need to know is that by the time we've gotten to 5 weeks, those 3 progenitor families of tissues have created the major functional parts of your brain. And you do need to know that by the 5th week, you have something that's called the diencephalon. You have the telencephalon, which is actually giving you the cerebrum, which is the cortex, which is where you do all of the processing that you actually do as a living organism in terms of cognitive functions. Definitely the frontal cortex of your brain is where you do all of your decision making. But for the purposes of anatomy and physiology, most of what we're going to be covering in this last week in terms of what's functioning where travels between the telencephalon, the diencephalon, and the myelencephalon down here, your medulla oblongata. And by the time we get through sort of introducing cranial nerves to you, we don't really go through what the cranial nerves do per se. We might describe that they're sensory or motor or mixed or something like that, or maybe tell you what modality of sensation they're collecting. But in total, we never really get through or get to how they all function. What's most important inside this class, definitely the way I write the exams, is where the cell bodies of the cranial nerves are going to be found. And the reason for that, of course, is that the cell bodies are where you actually instigate some form of a response to stimuli. And so it's going to be important that you understand that a series of cranial nerves, their cell bodies are found inside the medulla, or a series are found inside the pons. Maybe 1 or 2 are actually found inside the midbrain as well. Now that's just sort of like an overview of what we're going to be doing. But so essentially we're going to go through how do you build a brain. Okay, let's draw a brain for you. All right, it's going to take us 2 seconds. Cerebellum, cortex, pons, medulla, spinal cord, and inside of here, we're going to have this thing we're going to call the diencephalon, and that's why we spend this time making sure you pay attention to the fact that you understand that there's these different substructures, and you'll see why we point that out to you, I hope. Okay. And here's a cartoon of the development. This is inside your textbook. It's table 14.1. And again, it's not so much that you remember all the big names. You can dazzle people, when and if we ever get to Thanksgiving, right, with the names that you've learned. But what I want you to pay attention to here is really what you're doing. You are designing a system where the nervous system tissue lines a cavity. You're literally making a fluid-filled space here. And what happens is cerebrospinal fluid will occupy these spaces both on the inside and on the outside to ensure that these cells get the nutrients, oxygenation, removal of waste that's required for them to function properly. So let's start with the major parts, and I've already drawn those for you. You sort of have that brain stem, which is a continuation of the spinal cord, which was yesterday. And we're going to move upwards, and as you move superiorly, first you encounter the medulla oblongata, then you encounter the pons, and then ultimately you end at the midbrain. And there are functional regions inside of each of these. We'll go over those as we go through the major parts of the brain. Then we get to the cerebellum, which is posterior and inferior to the cortex itself. It's the second largest part of the brain. And for the purposes of our class, we think of it as the accessory structure for the regulation of movement, the fine coordination of how movement will actually occur. The diencephalon, nudge, nudge, hint, hint, wink, wink, is where we spend a great deal of time thinking inside this class. And what I mean by that is, what is the purpose of the thalamus? Why have we seen the hypothalamus since essentially day one? And you'll see it all the way through AMP2. And then we talk about this cute little part of your brain that's called the epithalamus. This is actually how we set our sleep cycles. And without sleep, of course, you can't get rid of the metabolic waste processes that I was just telling you about. So the epithalamus, even though I call it a cute little part of the brain, it's responsible for making sure we actually have normal brain function. And like I said, the cerebrum is the largest part of the brain. And by largest part of the brain, what I mean is this, is that during the development of the brain, it's actually a fairly smooth structure. And then as you grow, and in utero, what I mean you grow the cells that are required for what we will initially, or we will eventually call the integration of sensory input or information input to generate motor output. All of that will create these series of convolutions that we're going to call gyri, and those gyri are then folded into an area and they're compressed inside your skull. Okay, so you'll see how this works. So here's an image straight out of your textbook. I'm going to figure 14.1, and here's the spinal cord there. Remember yesterday, this is denuded, which means it has none of those meninges around it. Remember, the meninges were the dura mater, the arachnoid matter, and the pia mater. We will do exactly the same thing in a minute, right? You're going to see that that extension travels all the way up. So in black, believe it or not, we're going to create a meningeal system that will surround all of this so that you'll have your brain bathing in CSF. So even though they've done a really nice job of taking away all those tissues, it's an artificial sort of way of looking at it. So we peel away the meninges, and this is what you'll actually see inside class. They've actually peeled away the pia mater off of this brain as well. So you actually have true cell surfaces for each of the neurons that make up this area. Starting down at the spinal cord, we move into the medulla. You'll notice there's this sort of impression here of where the cell bodies of the neurons are inside the medulla. As we ascend and move anteriorly, you're going to see here is the pons. The pons will do many things inside of our body, but primarily for us, we're thinking about how it's going to be a relay point for information that ascends and descends. We have the midbrain, and sort of put this little chunk here. Okay, and that's the midbrain. You'll see it's responsible for being a relay center as well. And then as we sort of pass by all of this posteriorly, here's that cerebellum. You'll notice it actually has several of the same characteristics we're just speaking about up here. We have these convolutions of tissue. These convolutions of tissue increase the surface area. But remember, our cerebellum is for the refinement of movement as you develop movement inside your body. Okay. Next, as we ascend, we're going to find ourselves inside this area that we called the diencephalon. And inside that diencephalon, you have the thalamus, the hypothalamus, and back here on the posterior portion here, that's where our epithalamus would be found, or our hypothalamus, right? So here's hypothalamus here, here's a thalamus here, and our epithalamus. And the way to, at least the way that I remember it, is the thalamus is this central structure. If you move anteriorly and inferiorly, you're now at the hypothalamus. If you move posteriorly, it's usually somewhat a little superior. That's where you're going to find your epithalamus. So that's how you're going to hopefully just orient yourself a wee bit, okay? And I don't expect you to remember it today, but the complexity of responding to stimuli increases as we move further and further up through these physical structures. So believe it or not, the last thing to have actually evolved with you, of course, is the cortex of your cerebrum. You can see here those folds I was just telling you about. So if these are the gyri, the spaces in between are all the sulci, okay? And This increases the surface area, so you can actually do all of that great thinking that you do. Okay. Now, all of this has to be protected, and it's going to be covered by the meninges as well, and it's going to have the same pattern where you have a dura mater, arachnoid matter, and pia mater. But the distinction is the dura mater now inside your brain creates projections such that we actually separate each of the structures of the brain, where the falx cerebri is going to separate the two cerebral hemispheres, the right and the left. The falx cerebelli will separate the two cerebellular hemispheres. So that underneath structure that's actually responsible for controlling or modulating the movements inside your body. And then finally, the tentorium cerebelli is going to separate the cerebrum from the cerebellum. And when you do your dissections in lab, you're actually going to remove all of these structures so you can see the denuded brain. So when you're actually doing it, you're going to see something else as well. Inside of these structures, Mother Nature puts the return blood vasculature. So all of your deoxygenated blood returns into sinuses that are found inside of these. That's one of the things I want to point out to you here. So we have a sagittal section of the brain. Of the skull itself, right? Here's your face, your facial structures. Here's the spinal cord. And of course, they've removed inside this image all of the structures of the brain. Instead, focusing now on those connective tissue layers, you're going to see here that this is the falx cerebri. It literally creates a membrane that descends down to separate the right and the left hemispheres. You can't see it as well here, but the falx cerebelli is doing exactly the same thing. And then this line traveling all the way around is the tentorium cerebelli. And of course, now what this means is you have these functional separations where the fluid that's moving into these spaces is responsible for making sure those cells actually get the nourishment that they need. I call it nourishment, right? It's a bath of oxygen, carbon dioxide, glucose, and lots of salts. So maybe that's your idea of nourishment, but that's what it is. And in blue here, what I want you to see, and the significant part, of course, is that an area of a sinus is an area that is an area of lower pressure, which means what Mother Nature did is she engineered in all of these areas of low pressure in connective tissue, which means they're not likely to be compressed. And because they're a sinus, because they're a low pressure, it means all of the waste products from metabolism in your central nervous system, okay, actually are returned here. They'll descend down, ultimately joining your jugular veins and then depositing into your subclavian veins to return to your right atria. So we need to create this we need to create this environment, right? And we do this by looking at that blood-brain barrier we saw way back inside chapter 12, where you used ependymal cells, astrocytes, microglia, and what they do is they're responsible for making sure that the blood-brain barrier stops materials from passing into that cerebral spinal fluid. Okay? Now, why would Mother Nature do this? Well, obviously, she doesn't want to spend time thinking about what these cells need to be doing versus what the other cells inside of your body are doing. And she designates anywhere between 20 to 25% of all of the oxygen, I should say, all of the blood coming right from the left ventricle go there immediately. The cerebrospinal fluid itself is clear. Anytime you get a tap and it comes out not clear, that sends off major little red alerts for everybody who's looking at your cerebrospinal fluid. And it's going to circulate through specific structures inside the brain, and we'll talk about what they are in a second. It has a major function to absorb shock. If you've ever fallen and actually hit your head on the ground, something solid, the fluid in this small space actually protects the nervous system tissue from actually coming in contact with that dura mater. But it's also, from my perspective, the most important thing, of course, is to remove the wastes from the blood itself.
049986255.
How do we actually make cerebral spinal fluid? Okay, thought somebody was asking a question there. I was like listening closely. Nope, I don't want to listen to somebody's credit card number. I might steal it. Okay, so what you're actually looking at here is the how of making cerebral spinal fluid, right? So if we're looking at this, you can see that there are 3 major locations where we make cerebral spinal fluid, but they all have one thing in common. They're named a choroid plexus. That choroid plexus is where those blood-brain barriers will be found. And you'll notice it sort of becomes this little purple line. Cerebral spinal fluid that's now made travels in a somewhat linear manner through the ventricles to subarachnoid spaces to where they will be filtered out, meaning the waste materials are filtered out at these structures that are called arachnoid villi. Once filtered out, this is what then travels back to be reoxygenated, and the entire system keeps going. Why does Mother Nature do this? Because she's got a lot of surface area to cover, number 1. And number 2, in doing so, she can't. Give me a second here.
I barely listen to my voice messages. That would have been nice. Is there another way, like email?
Let me see. Something. Sorry about that, guys. Sorry.
Oh, okay.
Trying to figure out how to mute all here without being rude.
Oh, my business is I wanted to catch up on my payments. I want to know like where I stand right now. I know I haven't made any payments in a while.
Mute all.
Okay.
There we go. Sorry about that. Okay, let's get back to talking about class stuff. Okay, sorry about that, folks. Cool. Whack. So let's think about what this actually looks like inside of the brain. So here's a major choroid plexus. Okay, sort of, and when you actually cut open the brain, this is what you're actually going to see. It's not going to look red. In fact, it's going to look fairly brown. Oxygenated blood has traveled to this space. You create those blood-brain barriers where the surface area using the ependymal cells will allow for the filtration to occur. And it opens up into an open space. In this case, when you actually cut into the brain, you'll see the open spaces, and we call these ventricles. The fluid movement, sort of just follow the purple line, right? Fluid movement will travel, and by diffusion, will move down pressure gradients to then surround all of the tissues. And you'll notice inside this cartoon, they're suggesting it travels across the surface of the spinal cord as well. They neglect to point out, of course, it's traveling actually down that central canal inside the spinal cord structure as well. It will all be collected and then returned to these structures that are called arachnoid villi. So here's an arachnoid villi right here inside this cartoon, and they look like grape-like structures. There's a better cartoon inside your textbook, but the purpose is by increasing the surface area here, the waste materials can now move down their concentration gradient, and now you can see they've fallen into those sinuses inside the meninges. All of that gets returned now to your venous blood return, where reoxygenation will occur. So you get rid of the carbon dioxide. One of the things I'd like to stress for you, even though I mentioned sleep is really important, yada, 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 blah, blah, blah. A paper came out in Nature this week demonstrating the rate of clearance of particulate matter. And of course, the particulate matter that you worry about in this day and age, of course, is the neural filament tangles that are made that we believe generate or progress towards what we call Parkinson's and all the sort of dementia related disorders. And during your sleep phase, what actually happens is your microglia exit out of the tissues having destroyed those filaments. And those that don't do it properly, meaning you don't get enough sleep, the microglia then get trapped inside your central nervous system, and the filaments get trapped inside there as well. The microglia look like they actually get away, but they leave the mess behind, and that's what causes the filament formation you see at autopsies with individuals with progressive states dementia. Okay, where are we going next? Well, we're going to build a brain. We're going to start at the base because evolutionarily speaking, the medulla is the most complex portion of your brain. It's required for essentially everything that we think about in terms of thinking. And I mean by that subconscious thinking. You don't think about your heart rate. Would you agree? You probably don't even really think about breathing all that much. Okay, you don't think about how it is you regulate the movement between breathing and swallowing food when you're eating. But all of these are regulated at the medulla. So damage to the medulla, of course, perils your life. Okay. Now, the medulla itself has a series of hallmark structures you need to become aware of. One of which are named the pyramids, and these are anterior structures, right? And you're going to see them inside this cartoon here. That they bulge at the anterior of the medulla, and what they are is they're tracts. These are cortical spinal tracts that pass from the cerebrum to the spinal cord. And this is going to be significant because inside chapter 16, you're going to be learning what the cortical spinal tracts are. But from their names, you can figure out what they're doing. They're traveling from the cortex of the brain to the spinal column. And because you have 31 pair of nerves, depending on what type of information needs to be sent, that cortical spinal tract will then exit at, say, thoracic 4 or lumbar 5, depending upon where it needs to go. The medulla is a common site for decussation for both ascending and descending tracts. What this means is information. Raise your hand if you're left-handed, please. Nobody. Anybody? No? If there's anybody left-handed out there, all of your processing is done, of course, inside the right hemisphere of your brain. All of the rest of us who are right-handed, okay, all of the processing is done inside the left part of your brain. This occurs at these decussations. So as the information ascends, your brain intentionally says, oh, I want that to go to the opposite side. And we'll process that in the hemisphere of the opposite side of your brain. The idea is that you live in this sort of three-dimensional space where gravity is involved, and over the course of the evolution of the tracks as they evolved, it allowed for an easier refinement of movement. Nobody can prove that. We can't go back, I don't know, probably 3 million years or so when all these tracks were being developed. Okay, but we know how they are built. Yeah, go ahead, Amy.
Is it a physical difference that you can mark between right-handed and left-handed people?
You mean as in like where the tracks decuss and where they map inside each of the cortices? Yeah. Okay. Yeah. Well, and the thing is, of course, is then you get into that sort of that realm of like phrenology, which was big in the early 1900s. If you got this shaped skull, you're going to be good at, I don't know, X, Y, and Z. The thing is for whatever reason, there's a percentage of the population where it looks like it's a wiring sort of difference. Doesn't mean it's better, doesn't mean it's worse. It's different, and you can literally, I was explaining, I think it was yesterday, but I don't really remember, how it is we figured out how all these tracks inside the spinal cord actually work. You can literally go in and dissect them out one at a time, right? So it's a painstaking labor, but when you actually map the electrical activity, it's generally the same across each of the cortices. So even though it may go to a slightly different area, it seems like it functions the same way. Okay.
I'm sorry. I'm back asking.
Yeah, it's okay. No, it's good.
Going back to what you said about Parkinson's disease.
Yeah.
Is that is it hereditary the way the other dementia is, or is it affected by what you like, where you are and what you do and how much stuff you're exposed to that your brain might have to get rid of?
Yeah, so the honest answer is I don't actually know anybody who knows the answer to that question, right? There's the first part, like we're living much longer, right? Which means the cells that would normally do this job have to keep doing this job for a longer period of time. So the microglia, which are responsible for clearing all of this out, simply their efficacy could be dropping over time. right? The other factor that people are only really starting to pay really close attention to, and that is just the environmental effects of how much smog is inside that person's environment versus how much of a specific type of material is inside their diet. The genetics of Alzheimer's versus Parkinson's versus any of these other degenerative diseases, right? It's omnipresent. It's always going to be there, but nobody has the ability to point a finger to one gene just yet and go, if you're expressing that gene, you have a greater likelihood of it. I mean, I do know the people who are working specifically on the mechanisms of Alzheimer's versus, say, the mechanisms of Parkinson's disease. And they're looking specifically at what's actually happening. What are the cues to get, say, the microglia there? So that seems to be independent of genetics. But the microglia are the cells that are removing, in this case, for Parkinson's disease, removing the materials. Parkinson's and Alzheimer's for that matter. So even though you would never group them together, it's going to turn out the mechanisms will be similar. Okay. That answer your question?
Yes, thank you.
Sure, no problem. Okay, so why do we actually have a medulla? Okay, like I said, you may not like this idea, but I could walk up to anybody inside this class who wants to sacrifice themselves today. Sarah, we're going to sacrifice you. We're just going to cut open your skull. We're going to scoop out your brain. We're going to go all the way down to your medulla. And what's going to happen is you're still going to live. You're not going to have a very varied existence. You're not going to be the center of conversation. This is for sure. But I do promise your heart will keep beating and your respiratory center will keep breathing, right? So these things can occur and allow for things such as vomiting, swallowing, sneezing, coughing, and hiccuping, right? And each of these, believe it or not, are reflexes. So if you think about what you learned yesterday in terms of creating a reflex arc and how it would actually function, that's actually what's happening here as well. And you might be thinking to yourself, vomiting is not a reflex that I want to spend my time thinking about, but think about that first time somebody brought you to a party and you hadn't imbibed a whole lot, and they're just like, shots, 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 shots. And you're like, yeah, woohoo, I feel great. And next thing you know, you're nearly dead in the corner and people are just sort of like waiting. They're like, they're going to die? They're going to die? And what's going to happen, of course, is that toxicity is going to actually cause you to vomit. And the reality, the hope is that you haven't imbibed that much alcohol that you can't have a projectile vomit that clears your lungs. What usually kills those individuals, at some hazing party or something like that, is they vomit, great. But the problem is there's so much alcohol inside their circulatory system that it causes an ineffective contraction of the musculature, and that liquid then gets pulled into their lungs. So what ends up actually happening is they end up drowning. So each of these are a reflex, and that reflex is run out of the medulla. Okay, it houses 5 pericranial nerves, and Hannah and Ryan can reinforce this. You'll need to know where the cell bodies of those cranial nerves are, because what's significant to understand is if each of these is regulating a specific piece of physiology, you want to know when you start thinking about how they're doing their job, what Mother Nature needs, what type of information Mother Nature needs to send there. And more often than not, that information is coming from the hypothalamus. Now the midbrain extends from the pons to the diencephalon. Hold on a second here. Wait a second. Cut out the pons. The pons. Brilliant, man. Okay. The pons here. Okay. Respiratory center. It's going to have a series of cranial nerves. It also is going to allow for the passage of what we're going to call the nuclei, the olivary nuclei inside the brain. And you're going to see, hopefully a little bit later on, if I didn't cut that out too, when we're actually thinking inside chapter 16, why specific axons of neurons travel through these areas, because in many instances they actually have to get to the cerebellum as well. But leaping to the midbrain here, It's the pons to the diencephalon. You're going to find a ventricle there. Okay, the cerebral aqueduct is going to be found there. The cerebral peduncles, which are the axons of the cortical spinal, cortical pontine, and cortical bulbar nerves. Again, chapter 16 stuff, but you want to know where they are when I start talking about it. And the tectum, which is actually two very important parts of your brain. And remember, you're a bilateral organism. I'm going to show you a really weird looking image in a moment. And if you've ever seen Venus de Wildebrand, okay, it's one of these like 30,000-year-old sculptures that's sort of like the piece of art history that demonstrates that we thought about our world in 3 dimensions. And you're going to see inside your brain exactly what Mother Nature was creating so you can actually see and hear the world. So the tectum itself also is called the corpora quadrigemina, and it contains 4 rounded elevations, 2 superior ones, which are called the superior colliculi, and 2 inferior ones, which are called the inferior colliculi. And to see this tectum, you literally have to remove the cerebellum. Okay, so that paints an image for you, I hope. And what we've done here is you're looking at the posterior view of the midbrain, and you'll see here these surfaces, the middle cerebellular peduncles. These surfaces have been cut so I can pull the cerebellum out of the way. And here is the tectum here, where you have your superior colliculi and your inferior colliculi. And now normally what happens is I'm talking calmly inside the classroom and nobody's paying attention because you've all fallen asleep at this point in time. And then I do something like that, right? Okay, where I pick up a textbook and I slam it down on the table and everybody goes, right? Yeah. And what's happened, of course, is I've activated your inferior colliculi on the side of your skull that is hearing that stimulus first. And then what happens is your superior colliculi will then, as your body is turning towards that sound, focus your eyes to where it thinks the actual sound came from. Now, the reason why I say this is 3-dimensional is because you have 2 ears on either side of your head, which means they get the information at slightly different rates. And that means as your head is turning towards the visual stimuli, it's also getting that information at 2 different rates. So your brain then has to take all of that input and create a 3-dimensional image of the information that's actually now coming in. So that tectum's actually kind of cool. If you think about what Mother Nature actually had to design here and actually have it work. You'll notice I haven't really spoken about these little yellow things here. I'm just telling you where they are inside the brain. They get their own treatment at the end of this chapter, okay? So even though you'll notice what's happening here, it's, say, cranial nerve 12, we're working up to the higher numbers as we ascend through the midbrain. There's a pattern there. So even though they have really low numbers, those are the most important cranial nerves, those ones at the top as we ascend. Actually, sometimes you can live without them. Okay, I'm going in the wrong direction there. Now, the midbrain itself has within it these areas, okay, the substantia nigra, okay, the red nucleus. It's a large area inside of this space. This is actually how you control subconscious muscle movement. Do I have anybody inside the room who, when they get nervous, their leg may begin to tick, I think it may begin to like swing wildly, or their eye begins to twitch? Do I have any eye twitchers inside the class? No eye twitchers? Okay. What's happening, of course, is your lack of control of subconscious muscle activity as a result of that anxiety or whatever is causing it is what you actually see. Okay. The red nucleus is going to help us control voluntary movements of the limbs. So damage to this means you actually lose the ability to voluntarily control the movements to your limbs. And you'll notice the midbrain contains cranial nerves 3 and 4. You hear that structure inside your textbook. There's the red nucleus there. There's the substantia here. And you really can't tell what's going on here, right? You're just like, so we put cell bodies in a specific location. And that's ultimately what you want to take away from this. Mother Nature decorates this entire structure with the location of specific cell bodies, remembering the cell bodies of those neurons are what's responsible for instigating or terminating an action. We don't care what the action is yet. We just need to make sure we see that Mother Nature has stratified these levels of importance. Now, this gets us to this little cool area here, the reticular formation. And the reticular formation literally begins early in the midbrain and terminates at the end of the midbrain so that we can do one thing and one thing only. It extends from the upper part of the spinal cord through the brainstem into the lower part of the diencephalon. And you create this thing that's called the reticular activating system. And you may notice that today's Thursday, right? And Thursday you probably had less of a desire to log in than you did on Monday. Monday you were a little bit fresher and you're like, oh yeah, I'll show you. This is week 3 and I'm gonna go kick some ass. But by Thursday you're like, it's tomorrow Friday. Do I really have to show up to class? Or maybe even better, like you're in class and you're just sort of paying attention and next thing you know, you're thinking about X, Y, and Z, right? And what's happened there is the reticular activating system is making a series of decisions of what you're paying attention to. Okay. Let me help you understand. Is there anybody who likes to go camping? Fantastic. Look at that. Two lucky participants, right? And think about the last time you went camping, right? And you were going to bed, you had stowed your food away, you tied it up in the tree, you made sure it was safe, right? And then you're about to go to sleep and you hear a twig break outside, right? You're just like, but maybe you're a seasoned camper, right? And you're just like, oh, that's nothing, right? But now think about the first time you ever went camping. The first time you heard something break out inside the woods, you're like, right? What happens here is depending upon which environment you find yourself in, your reticular activating system changes its attention level to that piece of information. Okay, so while you're trying to go to sleep inside the woods, you'll notice that mosquitoes that you rarely ever heard at home sound like there are helicopters coming in trying to suck all of the blood out of your body. Okay, because you know you're in the woods, you know there's no way you can go home and get vaccine. Okay, your brain is like, uh-oh, these things are trying to kill me. Okay, so that the reticular activating system does this for everything. So when I do something like nudge, nudge, hint, hint, wink, wink, okay, during a lecture, what do? You tend to pay attention to that more, just a little bit more, because I'll bet if we tracked this over the course of the semester, you didn't notice what I was doing. Then you're like, oh, I know what he's doing. And then you're like, I mean, I can't tell what those nudge, nudge, hint, hint, wink, winks mean. So you stop paying attention to them. So for everybody who's not paying attention to the lecture right now, okay, nudge, nudge, hint, hint, wink, wink, reticular activating system. Okay, cool. Cerebellum, okay, the second largest part of the brain. And this is really cool because in the course of your lifetime, there are now 5 documented cases that I'm aware of, right? That through fMRIs or MRI imaging, different people have arrived at hospitals throughout the world with unregulated limb activity. They can walk, they can live, they can do something, but they're not functional the way you would normally think they're functional. Most people think they suffer a traumatic brain injury or they have some developmental disability, but what's happened is it turns out they don't have a cerebellum. Okay, now I want you to do the math here with me quickly, right? I said 5 documented cases, and this has been in the last 4 years, right? Out of 7 billion people. So think about how rare this actually is, where when you actually image their brains, they have no cerebellum. Now, nobody's ever thought a cerebellum is required for existence, but we've always thought that you wouldn't be able to move. And it turns out what happens is the refinement of their movement is lost, but the developmental piece of their cortex in their cerebrum compensates for the lack of the cerebellum. So it turns out cerebellum, you're not needed, but for us to actually pass our sobriety test when you get pulled over, right? Think about that for a second. Like you gotta put one foot in front of the other. You gotta touch your finger to your nose or something like that if you've never had one done to you before. Not that I have. I mean, I haven't. I just happen to have 2 police officers in the family, right? So exactly, right? So the idea, of course, is the idea, of course, is your cerebellum is what's responsible for making sure that you control subconscious aspects of skeletal movement. And one way to think about it is this. Is there anybody inside the class who plays a musical instrument? Okay. Well, and the idea, of course, is what you're doing is you're manipulating small numbers of musculature to what your auditory system actually hears. Right? You know what the pitch is that you're aiming for. You know the note that you're supposed to hit if you've read the music properly. And what happens, of course, is now your body needs to make sure that that happens. It also is responsible for maintaining your equilibrium and your balance. Remember, you're an organism. Yay, you're an organism. See if we can draw one of you guys today. Look, stick figure people. Woohoo! You're an organism that is standing on a planet. Right? Standing on a planet that's spinning on its own axis while traveling around a sun, right? And that means that you actually have to have, number one, the ability not to fall over while this is spinning, and you should be able to orient yourself relative to where the sun is at any given point in time. And that's what your cerebellum is actually trying to do for you, okay? You can actually, remember all those handstands you guys said you were going to teach yourselves how to do? How many of you even remembered? You're like, oh, that's right. I was going to teach myself how to walk on my hands. Long gone. But if you're actually trying to do that now, you're like, no, I'm just trying to open up the book and read at this point in time, right? But now if you put this into context, let's turn you over, put you on your hands. Here's your hands. Here's your arms. Oh, come on, Jonathan, you can do this. There's your head. This is gonna look weird. Sorry, folks. And there are your legs. Okay, now all of this is changed, which means now that your cerebellum now needs to figure out what physical equilibrium and balance actually look like relative to you now being your head actually being closer to the center of the planet. Okay, you don't tend to think of it that way, but those are the functional forces that actually have to be worked with, and that's what your cerebellum is actually looking to accommodate. Here's the structure itself. You have these folds that are called folia. Do you get that? Folds that are called folia. Yeah, it's their name, really. I'm not joking. Folia, look. Okay, and you have the white matter inside the central nervous system, of course, are the myelinated structures. So it means that the folia are unmyelinated structures surrounding the myelinated structures. And this was the exact opposite of what we saw inside the spinal cord, where the unmyelinated material is inside the central part of the spinal cord, and the myelinated portions are on the outside. So you can see here, you have 2 hemispheres to the cerebellum. They are attached by this central structure that's called the vermis. And then remember, we will put the meninges around this and then separate it from the cortex of the brain. Now the cerebellular cortex is made up of gray matter that are called folia. The arbor vitae is the white matter. We look back here. This right here, that's called the arbor vitae. Okay, and the peduncles, there are 3 pairs of these, right? They attach the cerebellum to the brainstem. Nudge, nudge, hint, hint, wink, wink. They're important because should these actually become damaged now, that means whatever they were inserting into in the brainstem is now not going to be able to function properly, which makes sense, but okay. Now the thalamus is a really cool part of our, yeah, go ahead, A. Go ahead, speak up. I can't hear you. I'm getting old.
Can you hear me now?
I can.
So I'm sorry, but I have a story that will help people with the cerebellum.
Did you shoot somebody in the leg?
I did not shoot anybody in the leg. My mother is a cancer survivor, and she had a stage 4 glioblastoma multiforme in her cerebellum. And they found it because she was unable to stand up without vomiting. And we thought she had the flu. They had to do an emergency surgery. They gave her a very bad prognosis, but it turned out that because it was in the cerebellum, it was her saving grace.
Yes.
And they were able to remove it. And now she's been in remission for like 10 years, but she does kind of seem like she's drunk most of the time.
Are you embarrassed to be seen in public with your mom?
No, of course
Good.
not. I'm just happy she's alive. But anyway, just if that helps anybody, I know it's going to help me remember all the stuff that
Yeah.
you just said, because I was like, oh, that makes so much sense, all of the things. So
Kick ass. Thank you for sharing. You know, the other day I was thinking about we should have asked some follow-up questions about your Tinder date. Like, what was this dude doing with a gun shooting himself in the leg, number one? And number two, wouldn't that be like a major, like, hey, I gotta go now, just get up and walk away. He shoots himself in the leg. That's just like a bad indicator. But,
look, I'm not with the guy.
that's not what I'm saying. It's sort of like, you gotta wonder what's going on there. But anyway, okay. Okay. So thank you. Thank you very much for sharing your mom's story as well, because I know that I had mentioned glioblastomas earlier when we were talking about the glial cells inside the nervous system stuff, and I'm hoping I wasn't indelicate when I mentioned it then. I just said that when you actually have these, it's the cells that are rapidly undergoing mitosis. So if they do transform, that's what makes that type of cancer so very aggressive. The thalamus. Take a look at this cartoon. Okay. Notice that your brain is color-coded, at least inside your textbook, right? And it's not, because when you actually open up these brains, you're going to see that there's no color coding on them at all. But what I want you to see is you can map the cortex of the brain to the region of the thalamus that is sending or collecting information from the thalamus. Okay, so the way to think of it is this. The ventrolateral portion of the thalamus, which is over here, maps to the red region right here. Blue, the ventral posterior portion, maps to the blue there. The purple, the medial geniculate, maps to the occipital cortex and the parietal cortex here, right? So each of these, when I say that there's a reason why Mother Nature has actually built the structures the way she's built them, is because ultimately you have to bring something to a central relay point, and that central relay point is the thalamus for sensory input. It is the major relay station for sensory impulses. Go ahead. Hit the back of your left hand. Wham. Okay? Go ahead. You're like, yay. Okay, so what's actually happened there is, if you've paid attention to what I've said so far, that sensation traveled up through the medulla, it decussed inside the medulla, and it now has moved to the right part of the thalamic mass and is now stimulating the blue region on the right hemisphere of your brain. Okay. That's how you want to tie this together. So I had you hit a specific part of your body, you collected a sensory input, it traveled up a series of tracts, ultimately traveling and decussing, first decussing and then traveling to that thalamic mass, to then, notice I told you, it was the blue portion, which means the blue portion, when you're thinking about a location inside of the brain now, all of your sensory input, somatic sensory input, is going to travel to this location. Okay? Make sense? Cool.
So your right hand, would it go to the other side?
Very good. Okay. Same thing. So, and what Mother Nature, there's only going to be a few exceptions to this. So when we teach you the exception, make sure you pay attention to the exception. We get to those inside the special senses. Okay. Now our hypothalamus, there are classes, seriously, when junior, senior year of college or grad school, where you spend the entire semester just thinking about what the hypothalamus is doing. And it's a series of nuclei that are collecting information as to how you would change the internal environment of the body using an endocrine response. Okay, so how is it? So imagine you actually have a certain volume of water inside of the body you need to maintain. Mother Nature has two mechanisms that are regulated through the hypothalamus. Okay, but the hypothalamus inside each of these nuclei, and you'll notice I am not having you learn the names of the nuclei. I'm just simply pointing out to you that each one of these regions has its own unique responsibility. So one of those regions, if we're thinking about water, would be monitoring the osmolarity of your cerebrospinal fluid. Okay, because if we filtered blood at the blood-brain barrier, the lack of water would also be detected inside the cerebrospinal fluid, and therefore the hypothalamus would say, hey, I think there's a little bit less water than we need. Let's get some information from your tongue. Is your tongue dry? The answer is yes. Are you sweating? No. Your body then makes a series of decisions to release 2 different families of hormones into your circulatory system to retain as much water in your body until such times as it gets back to the normal osmotic range. And that's the way your hypothalamus works. And that's why in the beginning, inside chapter 1, when we say that your hypothalamus regulates metabolism such that you maintain engineered equilibrium inside of your body, regardless of the material we're thinking about, let's say temperature, okay? Temperature is not even a material, right? Heat dissipation, okay? Your body can withstand a temperature range. And if you get outside of that temperature range, for you to become too cold or you become too warm, the hypothalamus will then relay a series of instructions to the rest of the body to now change this. Now notice a moment ago I said primarily those responses are going to be endocrine. That's regulated through the pituitary gland here, okay? But there are moments when, say, you have to increase your respiration rate. So then a signal will travel down to, say, the medulla to change the respiratory rate there, or to the pons to change the respiratory rate there. Above our pay grade at this point in time, but the significance of the hypothalamus can't be overstated, okay? You really, not so much remembering what each of the regions are doing, but the hypothalamus is where we regulate normal homeostasis inside of the body. It's also a place where we regulate emotional and behavioral patterns, eating and drinking, body temperature and circadian rhythms in conjunction with that epithalamus. And a way to remember this, of course, is, I think I asked this on the first day of class, what do you become when you don't eat for a period of time, right? Some people mentioned they became angry and stuff like that. Of course, what's happening there is your emotional disposition is changing and your behavioral patterns are changing because there's insufficient glucose inside your bloodstream, right? And now your body needs to regulate that. So it diverts energy to change your behavior to make sure those physiologic needs are taken care of. The epithalamus is that sort of cool place inside your brain, okay? It's small regions superior to the thalamus. It's actually posterior and superior to the thalamus. It contains the pineal gland, which secretes a specific hormone, and that hormone is called melatonin. Now, everybody inside the class who isn't sleeping, and they're like, my doctor told me to take melatonin, right? And of course, what it does is it allows the body to rest because it works antagonistically to serotonin, okay? So you woke up this morning, right? Let's think about you waking up, okay? What happened? Well, the sun came up, right? And your eyelids were, of course, really thin, so some amount of light actually got into them, and that actually creates serotonin production inside your brain. That's what actually wakes you up. As night progresses, what happens is less serotonin is made, and now more melatonin is made. And what happens so is you increase the concentration of that melatonin. That's what actually puts you to sleep. Now, some people don't sleep, right? Maybe you're like, you wake up at 3 o'clock in the morning thinking about all the chapters you haven't read yet or something like that, right? Okay. The idea, of course, is at some point in time, there'll be other stimuli that will be over the concentration of melatonin, which would then override your ability to actually sleep. Lots of sleep disorders being discovered these days. We just live in a really effing stressful world, right? So the most you can do, the best you can do, you know, put your shades down, close your door, I don't know, put on some white music in the background, but sleep. The seat of your intelligence is found on the top of your head. Okay? So, which in and of itself just sounds weird. Okay? Right? It's the cerebral cortex, and it's the gray matter of the cortex, where literally the neurons are processing electrical activity to figure out the significance of whatever that event is. Right now, you're trying to scribble down words or pay attention to what I'm saying, and what's happening is that information is traveling to a unique location, and that unique location, a decision has to be made. Important to listen to, unimportant to listen to, right? But seriously, that's all it comes down to, right? And you're like, oh, that's important. And then, of course, the moment you say, oh, that's important, your brain has forgotten everything else you thought was important a moment ago. So it's actually this sort of weird decision-making process that your brain has to go through. And what it does is it creates areas where it's likely to remember that experience. And that's probably the best way to think about this. Likely to remember. You can open up your skulls, right? I'm going to take Caleb today because Caleb's just looking too chill, right? And we're going to keep him awake. We're going to shave his head. We're going to cut through his skin. We're going to cut through his scalp. We're going to cut the meninges out of the way. And what we're going to do is we're going to electrically stimulate different parts of his cerebral cortex. And what Caleb's not aware of yet is whatever is being excited, I'm going to ask him a question and say, what memory did that bring forward? And Caleb will tell you something he did when he was 3 years old, when he stubbed his toe on a Sunday afternoon and it was raining out and he could smell the smell of dirt and the worms. And you're like, how the hell do you remember all of that? You remember all of that because those are electrical events that are actually stored. And the physical act of actually applying electrical charge to that area of the brain is what now makes that information available. The only way you actually access information inside your brain repeatedly is to practice accessing it, right? So when I teach you in a day or so, no, it'll be next Monday, right? No, it'll be next Tuesday. Your first order neuron will collect sensory stimuli, which will then access your second order neuron in the gray matter of your spinal column. It will ascend and then decuss inside the medulla oblongata, synapsing with your third order neuron in the thalamus of your brain, ultimately traveling to the primary somatosensory cortex of your brain. You got about the first sentence into that, right? And then after that, you're like, right? What ends up happening is you literally create a pathway of electrical information, and that reinforcement of that pathway of electrical information allows you then to layer on more and more information. So if you've ever wondered why I draw these stupid little cartoons, it's because you can always remember a stupid little cartoon, right? Your brain handles this really easily. But if I try to draw in the perfect eye, or the perfect smile, your brain is like, what the fuck is going on there? There's no symmetry. You get distracted and you miss the point. The point was, this is an incorrect anatomical position, right? Yes, no, maybe. Did anybody catch that? Okay. Now let's put it back into correct anatomical position. Arms down, feet down, right? And the palms pointing outward. Yay. Fantastic, yada, yada, yada. Of course, they have one arm that's longer than the other, but your brain catches a whole series of these pieces of information that are subconscious, and they will distract you. But so you actually have to practice putting together. That seat of intelligence, of course, is just the refining of electrical events that are occurring inside your body. Oh. Anybody ever actually have to like memorize a poem for class or something like that, and then have to get up in front of the class and sort of then have to spew it all back out? No? Microphone, Amy.
My other degree is in opera singing.
Fucking ass.
So I had to do that all the time.
I hope that didn't hurt too much. Right?
It hurts. It hurts every day.
Oh, wow. Right? What, the degree in opera or the sound of my voice?
No, the degree in opera. Your voice, I'm totally used to.
I was like, what were you thinking? But yeah, it's beautiful. But at the same time, You know, and your brain knows when you're not hitting the right note. Yes, no, maybe, and it distracts you.
You hope it does.
Yeah, okay. Now, how Mother Nature partitions where that information is going, well above our pay grade. But you've already been given a hint as to how that's all going to work. Pay attention here, right? That hint is all of your somatosensory input is going to travel to the primary somatosensory cortex here. So regardless of what I'm saying and how I'm saying it inside class, all that information is going to travel here, okay? And once it's traveled there, now Mother Nature needs to partition it as to literally your frontal cortex to go, what do I do with this? Okay, with me?
What do you mean by partition? Yeah,
it has to make a decision as to what to do with the information. Okay, what type of information is it? How important is it? And in, believe it or not, going all the way back to when you first started to learn, like once upon a time I told you the cruelest thing your parents ever did to you was what? Anybody remember this? No? They put a mobile above your crib? Okay. Why is that the cruelest thing they could ever have done to you? Because every adult looks at it and goes, oh, look, it's shiny and it moves. That would be so interesting. Well, when you're a mass of protoplasm who can't move their fingers and their toes, let alone reach for something, all you have is something that's just creating frustration for you. You're like, right? That's really what it represents to your central nervous system. Now, maybe the people who made the mobiles were like, yeah, we're going to fuck those little kids over. And maybe that was their real goal. But the reality is this, is that motivation to reach out towards the mobile becomes a pattern of electrical activity. And then, of course, if you're anything like Ryan, right? Ryan probably reached for it, and then he realized, oh, this gets me to the crib bars. And then he's like, I got two hands on the crib bars. Next thing you know, reaching for the mobile gets him closer to the top of the crib. And where did Ryan end up? Crib floor, like the floor of his big room, right? When he crawled out of it, aiming for the mobile. And the parents are like, what are you doing? And Ryan's going, I don't know what I was doing. I was reaching for the mobile, right? Yes, no, maybe. Okay, so you have all sorts of unintended consequences. So here's your brain. We cut into a piece of your brain, okay? And you can see that we actually have these areas that are physically separated. But when you look at a brain, you're figuring out the difference between the frontal lobe and the parietal lobe. There's this clean line here. Not so, right? When you open up the brain, you're like, oh, that fissure looks like it could be a little bit deeper, or that sulcus might be deeper. But generally speaking, you have a frontal lobe, parietal lobes in blue, occipital lobes in green here. And on the right-hand side of this brain, you have the temporal lobe. And then, of course, you have the cerebellum here. Each of these are going to have, they will house electrical events that will be stored there. Okay, and as long as you're alive, there's some continued baseline electrical activity happening here all the time. And even when you're barely awake, what's happening, of course, all of this needs to be regularly activated. 4 lobes, I just told them to you. Central sulcus is going to, let's see, go here. We are going to go back here. A central sulcus is right here. Remember I said it's not artificial. There is actually a sulcus, but its location inside every single one of your brains is in a slightly different space, okay? You have a precentral gyrus and a postcentral gyrus. These are the names that we give to the primary somatosensory area for postcentral and primary motor area. Think of precentral gyrus as that red strip. Postcentral gyrus is that blue strip, okay? In fact, why don't I show you that again to make sure you get that in your memory. 3rd time's a charm, they tell me, right? Okay, so the blue is the primary somatosensory, but is now called postcentral gyrus. Red, primary somatomotor. It's now called the precentral gyrus. So you have at least 3 different names for this one structure. Okay? But how does it all work? what's actually going on here? So here's a brain. Okay, it's been mid-sagittally cut, and they've removed the gray matter. So they've taken away all the thinking bits. And this is actually, yeah. So what they've done is so remember when I said you actually have a processing center. So if the way this works is sensory input travels to the thalamus, and we're traveling to the primary somatosensory cortex, which is somewhere right around here, all that information now travels in, and it has to go someplace. Like, what do I do with this? And what happens is Mother Nature creates three sets of fibers inside your central nervous system. You have commissural fibers, you have projection fibers, and you have association fibers. Commissural fibers are the easiest to remember. The corpus callosum, which is this large ribbon structure, these are all axons of neurons. And this is actually how you send information between the right hemisphere of your brain and the left hemisphere of your brain. Okay. And what this means is the rate at which information is sent is dependent upon the size of the corpus callosum. And unfortunately, gentlemen inside the room, our corpus callosum are all significantly smaller than the female's corpus callosums inside of the room, right? And it's thought that, of course, the corpus callosum for females is larger because they have to take care of the offspring, right? But there's nobody who's proved it one way or another. That's just sort of like babble. But the physical difference isn't babble, okay? The corpus callosum is measurably larger in all females relative to males. Now, what Mother Nature will do is if information needs to stay on one hemisphere of the brain, she will use association tracks. So imagine information arrives at the primary somatosensory cortex, and it will then travel to the anterior decision-making processes here in the anterior portion of the brain. It'll use association tracks to do that. Literally, it's like getting on the subway. Electrical subway, the information is now sent up here, and they're like, hey, what do you think of that? And you're like, well, this doesn't fit with my mental model for making decisions about fill in the blank, okay? In those instances where information has to ascend or descend, what she uses is she actually will use the, let's see, where are we here? Point it out to you. I'll find one for you. Projection tracks here. So sorry, your images were in the way. So projection tracks will be what's used to have information ascend and descend inside the actual brain itself. So now we have three different tools. Information can ascend, travel to the primary somatosensory cortex. Then it may have to either travel to the anterior portion, so it would use the same association or use an association track. But maybe it has to pass over to the left hemisphere. And in doing so, it would have to use the corpus callosum, and those would be commissural tracks. That's a lot. How are you guys doing? Brain's almost full. Are you excited to learn more things? Don't worry, we could. I'm glad, because we haven't reached our stopping point yet. But if you have questions, I would like to pause here and go, are there any questions? In doing so, allowing your brain to sort of settle in.
I had a question.
Fire away, man.
Circling back to when you wake up, like you start producing serotonin.
Yeah.
Isn't your RA, like reticular activation system, being activated too? Isn't that what wakes you up?
Yeah. So what that would mean in conjunction, Ryan, you might become aware of the fact that your foot is hanging out of your blankets and your foot is cooler than the rest of your body. And that's what they mean when your textbook says that it sums these pieces of information. But the awareness inside of your body begins with a chemical change of the serotonin and then the stimulation that is detected as a result of the activation of the RAS.
So first chemical, and then the RAS kind of like makes you conscious of like what's going on.
Exactly. And the point about the serotonin, of course, is your eyes are designed to have a thin palpebrae, that lid right? It's designed to be thin so light can pass through that readily. And that's how your brain knows when the sun is supposedly up or not. Okay?
Huh. Very cool.
Cool. Okay. So getting to if we have all of these things happening at once, if we have commissural fibers, projection fibers, and dissociation fibers, how the hell do you regulate all of this? And to do that, we have the structure that's called the basal ganglia. Okay, and that's basal ganglia, getting back to you being inside the crib, right? Not literally you. Maybe you were never inside of a crib. Like, I have no memories of being inside a crib, but that could just be that it was all just one big post-traumatic stress disorder. So you just sort of like blank it all out, right? But you have these 3 nuclei that are deep inside each cerebral hemisphere. We call this the basal ganglia. Okay. That basal ganglia is made up of 3 parts: the globule pallidus, the putamen, and the caudate nucleus. And this is where you actually initiate and terminate movements, okay, that are sometimes unwanted or sometimes needed. Your basic muscular tone, sitting where you're sitting right now, is being determined by the basal ganglia. And it's not just the physical tone of your body, it's also, say, the smooth muscle tone of your body as well. And how can this basal ganglia function? Okay. So it sits inside, so in the purple portion here, these are the 3 pieces of it, and it sits inside these locations where what it's going to actually do is work in conjunction with the limbic system. Every single one of you have a limbic system and you hate it, right? Literally, you hate it. There has to be something that pisses you off every single day. Have you noticed that? There's one thing that's sort of like, wow, I have an emotional response to that, and that's your amygdala. Your amygdala is trying to set the baseline for rage. So that's why we get this class out of the way early in the morning, right? AMP1 students tend to be a little bit more volatile than AMP2 students, right? So you're more likely to lose your temper. So if you get that shit out of your way during the beginning part of the day, the emotional part of your brain is now having hopefully less of an effect. That makes sense. So between the hippocampus, which is where you are assembling all of your short-term memories right now. If I said, what did I start the lecture off with? Most of you wouldn't be able to tell me, right? Because the short-term memories have been turned into little flashcards, and those little flashcards need to now travel from the hippocampus to the cortex while you are sleeping. Okay, so all that information you're trying to pile inside of your head, okay, while you're sleeping, what happens is that short-term memory is now sent over to long-term memory while you're asleep. And of all of the memories that you actually initiate, those that are associated with a smell are the strongest. And let me ask a random person here to see if they're actually in class or if they're. Let's go with Nicole. Are you with us today?
Yes, I am.
Kick ass, Nicole. Nicole, can I ask you, have you ever imbibed a beverage that made you throw up?
Not like literally throw up.
How about something you told yourself you would never drink again? Like you had this one horrible night with tequila. Have you ever had an experience like that?
Not tequila, but there was like this one drink that I ordered once, and it just tasted like cleaning supplies.
Sounds like a martini. Anyway, right? Yeah, so it's probably like pure gin or pure vodka or something like that. Now, my question for you is that, so the reason why I asked if you threw up or not is, if you smelled that drink again, would you ever drink it?
Oh, hell no.
Very good. Okay, so now I need, let's see, Melissa, are you in class today?
Am I what?
Are you great? Are you with us?
Yeah,
Okay. Do you feel like playing along today?
I can try.
Kick ass. So Melissa, can you identify a drink that had made you vomit before?
I don't vomit often. Like, I don't.
Well,
I
I'm not
rarely-
asking if you're a drunk. I'm asking you like, did you go out one night and you have like 3 shots of blackberry brandy and you just will never drink that again?
No.
No. Is there anybody in the class who's experienced this?
Me.
I have.
Yeah, definitely.
Okay, cool. So somebody who said yeah, definitely to that. What was the drink?
It was gin.
Straight gin or just
Straight
gin and tonics?
No, it was straight gin. I'll never have straight gin again.
I would never recommend it to begin with, right? So now,
Well,
Ryan,
where were you?
brace yourself, okay? Brace yourself. Ryan, what does the smell of gin do to the sensations in your body right now? Try and conjure up the smell of that gin.
Can I give it a try?
It makes me kind of
Yeah,
like
go.
my hands start sweating a little bit, and like.
Who is the other victim who wants to participate? Go ahead.
So I had Bacardi Dark. And I had I think I had chicken Alfredo that night. Right.
Oh no, I can tell where this is going.
So I had decided to try it because it was in the liquor cabinet. So I had took I think like 2 shots and I just threw up everything. So now it's like when I smell it, my body gets all tingly and then like my hairs on my arms will just like stick up and I'll just be like, no.
And that's your limbic system trying to keep you alive. How do you feel about that?
Pretty good. It's saving my life.
Yeah, well, in many ways, right? But the idea is this: is by coordinating those emotional responses, and you may not necessarily think of them as like pleasant or unpleasant. The idea, of course, is your body needed to create a system to detect when a bad thing had occurred to you. And now, when you smell that, you immediately instigate that entire response system again. It's kind of cool if you think about it. Okay. Okay, let's get to this. Oh, here's your limbic system. How do we actually organize the cerebral cortex, right? So this is the stuff that you find inside your psychology classes. We don't tend to spend too much time thinking about this, right? But Mother Nature, like everything in the world, needs to be organized. If you think about like a zip code or something like that, you need to mail a letter, you put the zip code on it to facilitate the letter getting to where it needs to be. By creating specific regions, we think about, say, the primary visual area. You have the occipital lobe, primary auditory area. These will be the temporal lobes. Primary gustatory area, the base, the postcentral gyrus, so on and so forth. I know I asked Ryan this question last year, so let's see if there's anybody else inside the class who has fallen, hit the back of their head, and actually seen stars. Is there anybody who's ever done that?
Me.
Was this after you drank the Bacardi Dark?
No.
Good.
I was actually trying to ride a skateboard.
Awesome. Great. And I mean, not great, but I'll bet the moment somebody says skateboard, there's a sinking feeling inside the pit of your stomach and you want to throw up again.
Yeah.
Exactly. So part of the reason why you feel that, of course, is what happened, of course, is you hit your head on the ground hard enough that your gray matter actually pushed into the dura mater and then pushed into the bone. And that actually caused all of those neurons to depolarize. And the stars literally are the depolarizations of the electrical activity of those cells. Okay? Now, because it was such an unpleasant feeling, when you actually gave yourself a concussion if you still feel like you're going to throw up, right? Okay? What actually happened is your brain just said, that's bad. You curled up in a ball for a while, right? And then your brain literally took time to actually heal that. Did you actually lose vision after this, or you just get up and like walk away thinking to yourself, you're never going to ride a skateboard again? Uh-oh, she's out throwing up.
Wait, repeat that?
Yeah, so did you try to ride the skateboard again?
No, I'm actually too scared to do that because I'm not a professional. So
Well, you don't have to be a professional to kill yourself. I'm not laughing at you. Believe me, I've cracked my head open so many times. I'm surprised I have a brain left. But
no,
the
I
idea,
actually gave myself a concussion for doing that.
yes, of course you did. Yeah, no, that's what I said. Yeah, the things you don't play around with, right? Gravity and your skull. Okay, it never works out well for you. Okay, not you personally, but you as in like the human race. Okay, and here's a mapping of all those different areas. Don't bother memorizing any of this. There's only 2 pieces of information you need to pay attention to here, right? Here's the primary motor area, which is called the precentral gyrus in red, and the primary sensory area here, somatosensory area, which is called the postcentral gyrus. And that gets us to this last cool piece here before we switch gears, okay? So this means we get to review. And you guys are like, review? How could we review? Well, I'll tell you how we're going to review. In your take-home quiz, I think I had you guys like draw a series of neurons, right, along the way. Woo! Right? You did find the take-home quiz, right? I'll take that as a yes. You're like,
Yes.
shit. Good, fantastic. Okay. Whee! Kick ass. Okay, so now imagine we have graded potentials happening here that might be depolarizing or hyperpolarizing, graded potentials happening here, graded potentials happening here, right? Action potentials happening alongside the axon terminal on each of these. Maybe not all at once, right? It's going to go from the first one to the second one to the third one. Now put all of this together and let's build a brain. And imagine what you're actually looking at now is the sum of all of the electrical activity within a brain. And this is why we describe a human being alive or dead as defined by their brain activity. Your heart may be functioning, meaning your medulla may be doing what it needs to do, but it may not actually be able to coordinate those activities in such a way that you can actually make it work. Okay, cool. Pay attention to the lecture, okay? But the idea, of course, is going to be this, is that depending upon the brain wave frequency, these all, in theory, monitor types of electrical activity that are specific for theta waves are supposed to, alpha waves are for concentration, theta waves are for relaxation, blah, blah, blah. I'm not sure you should take any of it as gospel truth, but the interesting thing is you can partition the type of electrical activity inside of the brain by filtering out what's going on, okay? And I know I had one student who's now finished his PhD. He's got his PhD in physical therapy now, right? So he's just finished it. And one of the ways he did really well in this class is he used what that binaural or diurnal beat music, right? Which helped him, he swore helped him focus while he was studying. Now, of course, there's a whole bunch of research out there that says if you can figure out a way to help yourself focus, it's something you should probably help yourself do. It takes more energy, but maybe that's one of the things you can go out and figure out doing while you're studying for this next exam. But that's pretty much where I wanted to stop today, I think. Next slide should be the
Wait, can you
cranial nerves,
expand on
right?
that more? The alpha waves for studying? What is that?
Yeah, so go do some work at looking at, music for studying, right? And you're going to find there's a series of research papers out there. Don't read the research papers, use the research from it. And you're going to find that there's music that's out there to help you focus while you're studying, and they call it diurnal or binaural beat music for concentration. I'm sorry?
Yeah, no, I just said got it. Yeah.
Got it. Okay, cool.
Makes
Yeah.
sense, yeah.
Yeah, so, another perfectly good hour and a half of your lives are gone. And I hope you never forget vomiting and remembering what your epithalamus does and why commissural association and projection tracts are so important. You know, the thing you're going to pick up tomorrow is why we need to learn where the cranial nerve cell bodies are located. Okay. And then after that, we'll review for your exam. I'm going to put up your online quiz probably this afternoon. I'm refining some of the questions, and that's probably going to have more to do with chapters 13 and 14 than say chapter 12, but I will definitely put a smattering of chapter 12 questions inside of that as well.
I have a question.
Yeah, fire away.
When you start talking about where the cranial nerves are located, do you have like, an idea on how to remember all of that?
Yeah, so people usually look for mnemonics and stuff like that. I am not a fan of them, not because I can't learn them, but because they don't actually add any value. You're distracting your brain to learn something else, like all women must do blah, 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 yada, yada, yada. You know, there's a table inside of your textbook I'm going to point you to tomorrow, right, that I think you should probably focus on that, and that will hold you in good stead, right? But the idea, of course, is how I remember them, okay, is I draw this. Here's a brain, right? Here's the pons, here's the medulla, right? And what I do is I go, so if here's the medulla, I go, which cranial nerves are here? Which cell bodies of cranial nerves are here? Which cell bodies of cranial nerves are here? And when you do that, you realize that most are found inside the central nervous system, and there are some that are not. Does that help?
It does, thank you.
Kick-ass. Any other questions? Nope. Well, I will let you back to your partially sunny day. I have a couple of fawns running around outside the window here. They're spotted, they're cute. Have a wonderful day.
Do you know what type of waves are emitted when your parasympathetic nervous system is being activated?
Theta, I believe.
When you're calm.
Yeah, theta waves.
Oh, okay.
Yeah.
Are these the same waves that you experience during your sleep cycle, like during REM and all that?
So the way they're modulated will change, all right? But yes. Okay. So remember when I was saying that during your hippocampus is responsible for shifting the information from when you are first acquiring it in short-term knowledge, and then it becomes long-term knowledge. What I mean, and what's happening there is you then shift which populations of waves become more apparent during, say, a specific sleep cycle. Does that help?
Got it. Thank you.
Cool, no problem. If you guys want to know more about that stuff, I can throw some of it in the lecture tomorrow, but it won't be on your exam. It's not like I'm going to ask you what do alpha waves do, beta waves, that's sort of stuff. What I did is I wanted you to understand how, if you have all of these different neurons and they're either being hyperpolarized or depolarized with action potentials, all of that, how you could get these different types of electrical activity. Okay, cool. Well, have a wonderful day. I will see you guys tomorrow. It's Friday tomorrow. Woohoo!
Woo!
Exactly. Don't start drinking tonight.
Almost woo, except for that when it's Friday, it's like, okay, quiz, test.
Yeah, it's not really a weekend.
I'm so sorry.
I know that you are lying. You are not sorry.
You could detect the change in my voice there. I must practice disguising him better. Yeah. Hopefully you started the take-home quiz now, because you don't want to leave that one till Saturday morning. That is for sure. Okay? A wonderful day. If you have questions, you know where to find me. Okay?
All right.
Yeah, be safe. Oh, man. Oh, end meeting. Bye.
All right. See you
You guys
later.
rock.